Basic research results 1990 to 1999. The following part sums up the results. In each case, the number of the publications listed in publications on the biological effect of DD water with the results and the place of examinations is given in brackets. Where there is no reference to any publication, results will be published in the future. 1. DD water 30 ppm inhibited the division of I fibroblast cells I, or SA goes oncology at National Institute of Oncology 001, Budapest. 2. The inhibition exerted by DD water was more expressed if L cells were synchronized by serum deprivation in phase GG, and hence transferred to deuterium-depleted medium I, 001, Budapest. 3. In nearly 60% of immunosuppressed mice with transplanted MDA and MCF7 human breast tumors, drinking of DD water caused complete regression I, 001, Budapest. 4. As a 11 effect of DD water 90 ppm, the tumor growth was slowed in mice with transplanted PC3 human prostate tumors, and survival rate increased by 40% in the treated group 6. Institute of Toxicology, Keste. 5. DD water 90 ppm inhibited in vitro growth rate of the A4 cell line murine hematopoietic cells. 5. 6. Patterson Institute, Manchester, UK. 6. The intracellular pH of A4 cells was restored with a delay after an artificial pH decrease in deuterium-depleted medium 90 ppm. The amyloid-sensitive Na plus H plus antiport system, which plays a key role in cell division, took place in the process Patterson Institute. 7. DD water 110 ppm induced apoptosis in mice with transplanted human PC3 prostate tumors. 5. 6. Semmelweis Orvostada Manjigietum Semmelweis Medical School SOT, Budapest. 8. DD water 90 ppm induced apoptosis in interleukin-independent A4 cell line in vitro. 5. Patterson Institute. 9. DD water 90 ppm activated the membrane transport process of the Cwe-Delodia quadcancy, causing an increase in pH in cells, and a decrease in the extracellular area to 1 and 11. Josephatilla Manjigietum Josephatilla University of Life Science JAT, Szeged, Hungary. 10. DD water 90 ppm. Simultaneously with the pH change, hyperpolarize the cell membrane in the Cwe-Delodia canadensis 11, JAT. 11. DD water 20, 60, 110 ppm, during germination, inhibited the sprouting of 10 different plant species 2, 4 and 8, JAT. 12. In the case of rice, corn, wheat, and squash, a deer dose dependence was observed, showing that the lower the D concentration, the stronger the inhibition of sprouting 2, 1, 8, JAT. 13. DD water inhibited DNA synthesis in vitro in 7 Monegasque Franks human breast, PC3 human prostate, and DIM14 human melanoma cell lines 1-5, Oncotech Incorporation, or VIN, USA. 14. Inhibition of DNA synthesis. In the case of 7 Monegasque Franks human breast, PC3 human prostate, and DIM14 human melanoma cell lines was higher and also lasted longer with cells synchronized in the phase G0G1 in vitro. 5, Oncotech Incorporation. 15. In the inhibition of DNA synthesis, a close dependence was clearly evident. The lower the D concentration of the culture medium, the stronger the inhibition. Oncotech Incorporation. 16. The application of DD water 90 ppm resulted in the total cure of a cat with lymphoid leukocysts 3, Alpha Velveterin Oni Hospital, Székesfehérvár, Hungary. 17. DD water 90 ppm resulted in significant tumor regression and healing in dogs with breast, rectum, and testis cancer 2, 4, 5, 6, and 7. Alphabet Veterinary Hospital. 18. DD water inhibited the proliferation of HT29 human colon tumor cell line in vitro but exerted a minimal effect only on healthy myometrial cells SOT. 19. The inhibitory effect of DD water was more expressed in the case of HT29 cell line 2. When cells synchronized in phase G0 G1 were placed in a culture medium with DD water SOT. 20. Inhibition of DNA synthesis was observed as a result of deuterium depletion in the case of tumors of the ovary and breast from newly operated tumor patients in vitro Neotech Corporation. 21. During experiments carried out with human tumors, the inhibitory effect of DD water doubled, increased to 40% in the experimental system, and inhibition duration also grew when the decrease in the concentration happened not in one step, 15,000, 20 ppm, but in 4 to 5 steps, 1506055514642 ppm in 2 to 3 days, Neotech Corporation. 22. Deuterium depletion, 20 ppm inhibited COX-2 gene expression in healthy myometrial cell line SL. University Medical School, St. Louis, USA. 23. Deuterium depletion, 60 ppm inhibited the expression COX-2 gene in the case of HT29 colon tumorous cell line SOT. 24. COX-2 gene inhibition correlated with the deconcentration of water. The lower the deconcentration, the more significant the measure of inhibition SOT. 25. The inhibitory effect of D-depletion on COX-2 also correlated with the prostaglandin concentration measured in cells. The more significant the inhibition of COX-2 expression, the lower the intracellular prostaglandin concentration SOT. 26. Deuterium depletion inhibited the expression of the HRAS and C-myconcogenase as well as that of the P.53 tumor suppressor V in MySi0. Pexi Orvostutum Foyagietum University Medical School Po, Pex, Hung.